I will do my speech in English. I hope it will not be a problem for you.
I want to thank you to the organization committee for inviting me to this prestigious event. Today I will talk about psychiatric diseases, modeling by organoids through stem cells. I would like to thank to my university, BAU Medicine, and also to my lab members, Somuncu lab members, for helping me evaluating this process. In the introduction section, I want to give a brief information about what is organoid and how to be made. Afterwards, I'm going to go with the fact that why do we need those organoids for disease modeling. And afterwards, I would like to talk about why we need them in cerebral studies. Afterwards, I want to give brief information about two gold studies processed in the world right now. One of them is published in 2013 in Nature, and the other is 2017 by Cohen. And also I want to talk about the procedures, the techniques that we developed in my lab, so what we do in the Somuncu lab, and organoid system optimizations and the cerebral organoids that we produced. Then I will open the subject of discussing it with you, hopefully. What are organoids? Stem cell field is already a breakthrough point for so many scientists, but after a while we realized that as stem cell scientists, we realized that 2D layer cell culturing is not actually mimicking the 3D properties of normal disease modeling. Therefore, we wanted to see whether the 3D model systems can be produced to mimic mostly other diseases, but then afterwards, once we achieve to have optogenetic analysis, we wanted to see whether we can do it in cerebral organoid systems and mimic schizophrenia, bipolar diseases, other mental disorders, etc. So organoids are in vitro derived structures that undergo some levels of self-organization and high levels of proliferation. And they have a resembling point, at least in part, and produce in vivo organs. An organoid generation relies on the remarkable ability of stem cells and the progenitor cells and their self-organizations once seeded in a hydrogel formation. This is a section through a cerebral organoid which reveals an organized neuron which was performed by Lancaster in MRC laboratory, which I personally know her because she is one of the leaders of producing organoids for the first time. This woman is Paola Arlotta. She is an Italian scientist working in the US right now. She has been the first woman scientist to produce the cerebral organoids for the first time in Harvard University. What she does is she has a specific diseased patient, can be both bipolar disease, schizophrenia, or can also be other psychiatric disorders. She does a reprogramming by using gene editing. In this current technology, we use CRISPR-Cas9 most of the time. Then once she does the genetic engineering, she starts aggregating those cells. Once she aggregates them, she do the induction of molecules, maybe growth factors or the cytokines, which will be needed for modeling the disease. And then they've done the maturation of organoids once seeded with a hydrogel. She is going to talk about her organoids for a while, so hope you enjoy it.
Typically, when you make a neuron or any cell type of the nervous system from a pluripotent stem...
Let me start from the beginning.
Typically, when you make a neuron or any cell type of the nervous system from a pluripotent stem cells, these cells retain very immature type of features. So what we have been able to do is to start from pluripotent stem cells and find culturing conditions that would allow the continuous development of these organoids over a longer period of time. And so this allowed the generation of a great diversity of cell types which were added over time, and also the maturation of the cells that are made was much more advanced than what was previously possible.
Before going further to my second slide, I want to say that it is such an opportunity to start working with the 3D mechanisms in Turkey, because so many labs in Turkey at the moment are still doing drug testing or psychiatric analysis by using 2D layers, which is normal cell culturing conditions. Therefore, we started to do it. Not only me, but also other institutes started to do 3D modeling systems. So we are really happy that it is progressing itself. So why do we need those organoid systems? This is the first point. In 2D conditions, you are not fully allowed to do a molecular mechanism analysis, which is basically lying behind the disease progression, and the identification of potentiality of novel biomarkers and the development of patient-specific platforms for drug testing or cytotoxicity analysis, and also bringing the promise of personalized medicine for each and individual patient. So this is why we need 3D systems, because in 2D systems the extracellular matrix components are not going to mimic your own body, or the growth factor cytokines or the hormones being released by individual cells will not be mimicking the normal human body. At the end of the day, even the animal models, since they are a little bit of different from us, is not fully complementing ourselves for disease modeling, unfortunately. Why do we need the brain organoids on the other hand? So organoid models provide an opportunity to model the unique features of human brain development in a complex tissue-like compartments. The second reason is the intrinsic patterning of whole brain organoids in intrinsic organoid protocols promotes the generation of diverse brain specifications, the regions. Third is signaling molecules can be directly used to mimic the in vitro and also sort of in vivo condition. And there are a number of advantages of an in vitro system for experimentation purposes: relative ease of genomic manipulation, heat map analysis, single RNA sequencing, protein expression levels, etc. Protocols will benefit from systematic analysis to cell types, so you can directly select your cell type of interest and seed them together to make a heterogeneous population. After a while, they will have the ability to migrate to specific regions to make a 3D model system for analyzing cerebral organoids. And pre-patterned region-specific organoids can be also fused to model complex brain structures. This was one of the best news for organoid researchers, which was published in Nature Reviews recently. So the cerebral organoid researchers realized that even if you seed heterogeneous population, meaning that glial cells, astrocytes, etc., in a hydrogel formation, even after a while, they are initiating to migrate to specific regions to produce this rosette formation, which can be able to mimic the intercellular communication inside of a cell. So even without doing any further touch from your own experimental process, even if you seed it by itself, they will be able to migrate and produce organ systems mimicking your own cerebral system. There are optimized protocols. If you are willing to work with the stem cells, in Turkey we are not allowed to work with embryonic stem cells in human therapies, but we can use it in vitro. Some universities are already doing it. So what they do is they are taking embryonic stem cells, or since it's not actually happening in Turkey, they use the induced pluripotent stem cell, meaning that they are taking fibroblastic cells, already differentiated cells from a patient, and put them gene transfections and then make them go back to the embryonic state to further mimic what is going on and mimic the disease properly. So what is happening is ESCs or the IPCs both getting together, and then the intrinsic factors and the extrinsic factors can be stimulated. For instance, for the forebrain you can add TGF-beta, activin, nodal inhibitors, IWRA and Wnt inhibitors to mimic the forebrain cells and make your embryonic stem cell or the iPS cells to differentiate into forebrain specific cells. There is also another publication also happened in Nature, saying that the cerebellum, dorsal cortex, ventral cortex, hippocampus and choroid plexus can also be mimicked by using specific inhibitors and the growth factors. So these two actually started this cerebral organoid process, Paula was the first woman who had the hypothesis of making the cerebral organoids, making 3D organoid systems, but these two were the ones who actually tried to employ it. So they are working, currently one of them is working in London. I had the pleasure to meet with her, and the other is working in IMBA, Vienna. What they do is they've taken embryonic stem cells or the iPS stem cells, then they've had the SOX1 and PAX6, which are specific markers for cerebral systems. They transfected them with those markers. After a while they've seen that they were able to make cortex. Then after a while they had this rosette formation even without touching the organoids, so they started doing directly experimentations on the cerebral system. What they do is mostly about cancer biology, not in psychiatry, but there are also studies doing psychiatric analysis by using the organoid systems. So this study has been published in Current Opinions in Neurobiology in 2017. What they have is optimized protocols for each individual differentiation process of different cellular types. So for dorsal forebrain neural progenitor cells, ventral forebrain neural progenitor cells, midbrain, hippocampal and forebrain neuroepithelium, they have different protocols, and especially they now optimize another protocol for turning embryonic stem cells into Purkinje neurons, which will give a clear idea about the disease progression in the future. After a while they actually used it in the protocol for pluripotent stem cell based modeling of neuropsychiatric disorders. They had neural rosettes. Once they achieved to have the neural rosettes, they mimicked the Rell syndrome, they mimicked PDS, schizophrenia, etc., and they have also mimicked the microcephaly reasons. So let's go back to the definition of what is schizophrenia for a while, because in order to understand what we've done in the lab, we need to repeat the definition of it firstly. So the NIH suggests that schizophrenia is a chronic and severe mental disorder that affects how a person thinks, feels and behaves. There are positive symptoms that we are all familiar of, which are hallucinations, delusions, thought disorders, movement disorders, etc. And the negative symptoms, which are the flat effect, having no emotions, no facial expression, and reduced feelings of pleasure, difficulty in beginning and sustaining the current morals. So after a while you have different stages of the disease progression. In our model system, we managed to mimic the exact time point of having an insensitivity to the drug therapy. You know that in risperidone therapy or etc., after a while using the drug makes the person, the patient, to get insensitive to get treated. So in that area should be mimicked to understand what's going on in the cellular level. So what is schizophrenia in the cellular level on the other hand? There is an inflammation factor and the immunity happening in schizophrenia, meaning that the pro-inflammatory cytokine expressions increases and the anti-inflammatory cytokine expressions decreases, which causes the cortisol levels to be increased and also the synaptic plasticity to decrease. And then we start to see the negative symptoms and the positive symptoms. Another reason is that the normal conditions, the activated microglia gets inactivated and turn into a resting microglia, meaning the systemic and the central nervous system inflammation leads to activation and the priming, which causes the T cell activation, meaning that it adjuvantly causes the B cell activation, meaning the neurodegeneration. There is also another current study saying that not only those are the reasons for schizophrenic disease development, but also the schizophrenia related, meaning that schizophrenic stem cells do not have the ability to directly differentiate into a normal functioning neuron. Even if you try it in a cell culture, they won't be able to highly proliferate to turn into a normal neuronal functionality. So that's what scientists try to do at the moment. Take the schizophrenic stem cells and then do CRISPR-Cas9 gene editing to make them further differentiate into healthy neurons and then transplant it back to the patient. There is also another reason that we are all familiar to, the brain-derived neurotropic factor, which may lead to schizophrenia or depression. So the serum BDNF protein concentration is significantly elevated in schizophrenic patients as expected. This is some of my lab members. In this study, I worked with BAU medicine students, and also I have a master's student coming from Istanbul Technical University, Pırıl. She's also helping me out to figure this out. The cerebral organoid studies of Somuncu lab, we can say. The first idea of the lab was the fact that we may be built on the idea of whether the glial dysfunction has a both similarity to attendance to both cancer glioblastoma and also schizophrenia. So the current therapies actually understood that some of the glioblastoma tumor patients, after they die, so the post-mortem studies showed that they were having schizophrenic treatment. So the schizophrenia treatment actually induced and elevated their life rate, meaning that for 5 years tumor clear, nothing's happened in the body by using the risperidone treatment. So this gave us an idea whether we can use directly the glial cells, the glioblastoma cells, to mimic schizophrenic models, to understand what's going on. So what we've done is we had different types of glioblastoma cell lines, U87 and also C6 cell lines, and we've done genetic screening to understand these cells are actually cancer cells, so are they going to cause some sort of high proliferation rate regarding to the cancer genes being expressed. And we realized that actually these cells having upregulated cancer gene expressions, but not that high, so they can actually mimic the system of schizophrenia once you model it. Since you're not going to transplant it to a patient, it can be a good system for mimicking the disease without using animal models. Then we've taken U87 and C6 cell lines and co-cultured them with monocytes which have the ability to turn into macrophages for immune system mimicking, and also feed them with endothelial cells, the angiogenesis promoting factors, which is going to come with the human brain endothelial cell lines. Then normally the organoid systems, the ones that Paula, Magdalena and Nüblich did, was actually using matrigel formations. Matrigels are really great hydrogels, which normally in room temperature they are frozen. In high temperatures they are melted, so it is the vice versa in normal life. But unfortunately they are really, really expensive. Like even if one vial is like $5,000 or something. So what is happening is we wanted to actually understand the matrigel is working. So we've seen that matrigel was working, but the proliferation was not as good as the literature suggests. So we said that let's produce our own hydrogels. Then we've used the agarose, normal agarose that we use for gel electrophoresis. We optimized the protocol by using different media conditions that I cannot talk about right now. And after a while we seeded the cells inside of that hydrogel formation and seeded them into a plate. After 3 weeks, as you can see, we had the cerebral organoids here. Once we had the cerebral organoids, we started to do drug testing. The drug that we tested for the first time was risperidone, because it is one of the actively used schizophrenic treatment drug at the moment. Then we've done the immunofluorescent analysis, the total bilirubin, total glucose, total antioxidant and oxidant therapies levels, and also the real-time PCR to understand what's going on inside of the cerebral organoid. We didn't do protein expression analysis because in our lab and in the world as well, since almost 60% of the disease modeling, the mental disorders happening regarding to the epigenetic factors, investigating the epigenetic mechanism should be another page that should be investigated. But in the first time, the genetic materials should be the first point to start this cerebral organoid studies. So once we seeded the cells, as you can see, even after the week 1, we start to see the spherical cerebral organoid formations under the microscope. But they were really tiny. And after the second week, we realized that the organoids getting bigger, bigger and bigger. And then we said, what is the mean value and the length value of those organoids? So we realized that U78 systems are really good for organoid modeling. Then we wanted to go with the positive control, meaning that the ones being used in the literature at the moment by different scientific groups. C6 glial cells were also attached to the hydrogel in good condition, therefore they weren't able to form the rosette formation as the U78 do, 87 do. So they had good mean values, but different from each other by p-value, and the length was cool, but they weren't actually producing good structures of the organoid. Then we had this idea, why did we go with risperidone? Because risperidone, as you know, is the DRD2 and 5-HT7 antagonist and have shown to suppress growth and progression of glioblastoma in schizophrenic patients. And also, they suggested that the patient's long-term survival from glioblastoma may be linked to the long-term risperidone use after post-mortem studies. So risperidone is hypothesized to have a potential anti-tumor effect and also anti-schizophrenic effect. Therefore, we further investigated the exact cell viability dose, the optimum dose for applying it to organoids. In the literature, normally it is 150 microgram per milliliter, but our results suggested that they were 205 microgram per milliliter was the suitable dosage for giving it to the organoid systems. Then we started experimenting it. Firstly, we checked the total glucose levels. So total glucose studies in schizophrenia, normally it was shown that the first episode relatively old antipsychotic naive patients with schizophrenia had higher levels of plasma glucose. And also once used risperidone, the levels get elevated further. Our results suggested the same. If your organoid systems are cool systems, then it should be having upregulation of risperidone used, of total glucose, and also upregulation of already existed organoids. The results were actually correlating with the literature. Then we moved on to see whether the total antioxidant and the oxidant status were correlating with the data. Normally the schizophrenia is accompanied by increased lipid peroxidation and nitric oxide levels and lowered antioxidant levels, which is expected. As you can see, in the total oxidant status, the organoid systems had it highly elevated, and once used risperidone decreased. And also C6 organoids showed the same result, but it wasn't the same for total antioxidant status, gave us a question mark saying that so the literature-based organoid systems are not effectively suitable for investigating it. But our system is better, we believe. Then we wanted to see the total bilirubin. So far, not so many studies investigated bilirubin levels in schizophrenia. It wasn't expected to have high differences in bilirubin levels. Therefore, we linked it with another condition. So in the bilirubin, schizophrenic patients have shown to have higher unconjugated bilirubin levels when compared with healthy individuals, which gives higher lipid peroxidation levels, meaning that active oxidant status levels. As you can see, it was highly significant that in the organoid systems, the unconjugated bilirubin levels were really high. Once risperidone used happened, the level decreased significantly. But it was the same for C6 organoids, meaning that again, the C6 systems are not efficient systems for disease modeling. Then we wanted to check the total cholesterol. The cholesterol level is upregulated in schizophrenia, but upper-regulated in risperidone usage. Again, our data was correlating with it, but the difference were not that significant as a p-value mean. Then we've done relative mRNA expression level analysis. This is not an mRNA expression level analysis. I just wanted to put that image because I really love the organoid. I just wanted to put it there. But since we start to look at the mRNA expression levels, meaning the real-time PCR analysis, normally in the serum levels of MMP2, metalloproteinase 2, the MD group which have depressive episodes, again the schizophrenia, significantly have lower than those of control groups. And the risperidone usage actually induces the Western blot, and the real-time analysis show that risperidone usage increases the MMP2 activation. So it was the same as expected in the organoid systems. Again, the C6 didn't give significant results. Then the interleukin-6, which is a really key point of inflammation, we expected to see higher levels of interleukin-6 in the disease systems. Therefore, they weren't that high. Once used the risperidone, they elevated. So it was basically the vice versa with the literature. So we had a question mark in this system, meaning that the organoid systems can mimic it, but at the same time, they are not inducing the inflammatory system that extensively. which is a really good system because you can mimic it by immune system cells seeded inside of the organoid. Therefore, you don't need to activate it once transplanted to a patient. Then the TNF-alpha levels was evaluated, tumor necrosis factor alpha. So comparison of TNF-alpha concentrations in patients before and after treatment of risperidone showed that the decreased level of TNF-alpha and increased level of interleukin-10. So the decreased level was actually expected once used the risperidone. But the C6 systems were again giving the contrasting results to us, meaning that they weren't efficient models for disease modeling. We've also done the TGF-beta expression levels and the insulin growth factor 1 expression level analysis. Both were correlating with it. The only thing not correlating with it was the TGF-beta. Again, made sense to us because they need to be correlating with interleukin-6. So we were saying that they're not causing inflammatory response. NLC3B, which is a really important marker for autophagy. So in the autophagy, it plays a key role in pathophysiology of schizophrenia as manifested by a 40% decrease in Beclin-1 mRNA expression. As expected, the LC3B expression in the organoids were really low, and once used the risperidone, both in the C6 system and U87, the expressions were highly elevated. Then we've started to do the good part, because observing the organoids under the fluorescent microscope is like magic. Why do I say that? Because you see the rosette formations under your eyes. So as you can see, the thicker areas like this are the rosettes that they produce without us touching it. So they can actually mimic the system. In normal conditions, we expect to have higher expressions of GAC, which is a really good marker for, again, both autophagy and lipid metabolism. And also, we've seen that once having an organoid system by glial cells, the expression was getting lower. Once treated with risperidone, the expression levels significantly increased for U87. Therefore, it wasn't the same for C6. The expression pattern was almost equal, so not giving a real exact result of significant result of having this organoids for GAC expression levels. Then, this is sticked. Then we've done the NF-kappa B expression analysis. NF-kappa B is a really important marker because NF-kappa B signaling is involved in neuronal development and the memory. Most recently, NF-kappa B activity has been reported to be significantly decreased in the brains of schizophrenia and increased once treated. The results were correlating with each other. Even in the C6 system, you see the expressions. It is highly, highly elevated. Then we've looked at PLC3 activity. The only singular reference study about PLC3 activity was the ones having that once the PLC3 is polymorphic, meaning that mutated, then you can expect to have different expression levels both in schizophrenic and healthy. So if the schizophrenic patient is not genetically herited, then you're not expecting to have differentiated expressions. Since we've done it in the lab and we're not expecting any genetic polymorphism, the expression levels highly correlating with each other. But in the U87 subjects, the expression was highly elevated, meaning that U87 can be an amazing model for mimicking genetic-related schizophrenia in vitro cultures. So, as a discussion points, we can say that C6 organoids, they are integrated with the hydrogel, but not as good as the U87 system. They were easily integrated with the hydrogel. Not efficient in 3D organization for neural circuits. They are better in rosette formation in neural circuits. Worse model than U87 system for serum media related in vitro analysis and colorimetric assays, and highly correlating with the literature. They had a problem in mimicking the disease progression for gene-based investigations. Therefore, both thought to be suitable models for the insensitivity phase, which mostly happens after 6 months of treatment. We can say that our system, which has been done by using the induced pluripotent stem cells, turn them into the glial cell, hypotized, is going to be the best option for disease mimicking for schizophrenia. I also would like to give a brief information about the symposium that I'm organizing. International Congress will happen in BAU Medicine next month. We all would love to see you there. The scientific schedule is already in the website, so you can check it out. Any questions? Thank you.